Good afternoon and welcome to a webinar from the Found Animals Foundation entitled Applying for the Michelson Prize. My name is Shirley Johnston. I am Director of Scientific Research here for the Michelson Prize and Grants Program at Found Animals in Los Angeles, California. And I'm joined by our Program Director, Katie Palfrey.
Thank you so much for taking the time to be with us today on this webinar. My name is Katie Palfrey. I'm the Program Manager of the Michelson Prize and Grants. And I'll start off with a brief overview of the Michelson Prize and Grants program, and then Dr. Johnston and I will launch into a discussion of the process for applying for the $25 million Michelson Prize. Found Animals Foundation was founded with the mission of minimizing shelter euthanasia of cats and dogs. Currently, 6 to 8 million animals end up in U.S. shelters each year, and 3 to 4 million are euthanized. In order to address this issue, Found Animals, in October of 2008, launched the $75 million Michelson Prize and Grants in Reproductive Biology. The program includes two components, the Michelson Prize and the Michelson Grants. The $25 million Michelson Prize will be awarded to the first entity to provide to the Foundation a single-dose, permanent, non-surgical sterilant that works safely and effectively in male and female cats and dogs. In order to fund the research to develop this product, the Foundation offers grants of up to $250,000 per year per grant through the Michelson Grants for promising work in pursuit of non-surgical sterilization for cats and dogs. Proposed research for the Michelson Grants is not required to generate results which meet all of the prize criteria in order to be funded, but preference is given to projects with the potential to produce a prize-winning product or technology. Grant recipients are eligible to make prize claims if research generates a product that meets all of the prize criteria, but you may also apply for the Michelson Prize without ever having received a Michelson Grant. The Michelson Prize and Grants is an international competition and is open to both academic institutions and commercial entities. So you may be wondering why development of a non-surgical sterilant for cats and dogs is so important. Surgical spay and neuter is currently the standard for pet sterilization, and while this surgery is very safe and effective, it remains out of reach for populations that drive shelter intake. Low-income pet owners cite costs and access as barriers to getting their animals spayed and neutered, and 95% of feral and free-roaming cats are unsterilized. The rationale behind the Michelson Prize and Grants is that a low-cost, non-surgical sterilant for cats and dogs could be used to address these populations of animals that are not currently being sterilized and which are driving shelter intake. The products would have a great impact on the number of animals ending up euthanized in shelters. Such a product is also likely to reduce the number of rabies cases worldwide, which are primarily due to feral dog bites. The program's goal is to bring a non-surgical sterilant for cats and dogs to market as quickly as possible, and to market this product at a low cost, so that it is accessible to the populations that need it most, including shelters, low-income pet owners, feral populations of cats and dogs, and NGOs in developing nations. For this reason, the Foundation seeks first right of refusal to an exclusive license for all products resulting from research funded by the Michelson Prize and Grants. Found Animals will fund manufacturing and commercialization of a promising product or products. In order to win the $25 million Michelson Prize, the investigator must show that the product is single-dose, permanent, non-surgical sterilant that is safe and effective in male and female cats and dogs, and can be administered in a field setting. It needs to have a viable pathway to regulatory approval and a reasonable manufacturing processing cost. But what do these criteria really mean? Here's the fine print. Single dose as defined as administered in a single patient encounter. For the product to be considered non-surgical, it must not require general anesthesia, but may require sedation or local anesthesia. For the purpose of the prize, a subcutaneous implant is non-surgical. In order to be considered permanent, the products must induce sterility for the reproductive lifetime of the species, defined as at least 10 years after administration. The Foundation will generally require 3 years of data with a trend line with no expectation of recovery. And though it is not a requirement of the prize, a product that eliminates the heat cycle in females is highly preferred. To be considered safe and effective, it must be one product for both species and genders, and it must be no more dangerous than surgical procedures. The sterilant must be safe for the animal, the environment, and the person administering the product. The foundation will require that efficacy is greater than or equal to spay and neuter. The product must not induce clinical abnormalities or pathological lesions at 1x dose in safety studies, and it must not be carcinogenic, mutagenic, or clastogenic. It also must not be a bait formulation. Finally, the last three criteria. It must be suitable for administration in a field setting, as defined as may require refrigeration, cold chain, but no frozen storage, water source, or other electricity than refrigeration. It must have a shelf life of at least 2 years, and it must not require a surgical theater. For reasonable manufacturing costs, the foundation defines that the target for a low-cost product is less than $25 per dose when manufactured at commercial scale. And applicants must have a clear understanding of the manufacturing process and costs, but don't need to have the capacity to perform the manufacturing. Finally, for a viable pathway to regulatory approval, there is no expectation that an applicant will have submitted for or received regulatory approval, but there should be a clear pathway. Regulatory approval from FDA CBM will be sought by found animals as a prescription product in dogs and cats. So how do you go about proving that you have a product that fulfills all the criteria for the prize? Well, you must first remember to always abide by the animal welfare guidelines throughout all research in pursuit of the prize. I encourage you to download these guidelines from the site today so that you can ensure you are in compliance. This is the process by which a researcher applies for the Michelson Prize. To win the prize, the applicant must generate data demonstrating safety and efficacy at a proof of concept level in cats and dogs, and the lifetime effect in a rodent model. The first step in the process you most likely have already done, which is to register on the Michelson Prize website. The registrant must then submit a registration letter which can be approved or declined by the foundation. If the registration letter is approved, the investigator signs the competition guidelines. The investigator should familiarize himself or herself with the data that will be required for data package 1 and may apply for a Michelson grant to develop this data. The researcher may also work independently at this stage if he or she has a different funding source. Once the researcher has the data necessary for data package 1, he or she will sign the master participation agreement and submit the data to the foundation along with a $500 application fee. If data package 1 is approved, the researcher will work with found animals to design a study for developing the data required for data package 2. A CRO will complete the study and submit the data to the foundation. If data package 2 is approved, the prize will be awarded. We've split the application process into 3 major steps, as you can see. Registration, which is the preliminary stage. Application, which is the development of and approval of data package 1. And the prize study stage, which is the development and approval of data package 2. Once the investigator registers on the Michelson Prize site, he or she may download the template for submission of the registration letter. The registration letter must include a list of the collaborators and indication of the person or entity that will receive the $25 million check should the group win the Michelson Prize. The registration must also include basic information about the proposed product, similar to what the foundation requires at the letter of intent stage for the Michelson grants. Additionally, at this time, the investigator signs the animal welfare guidelines. Members of the Scientific Advisory Board will review the registration letter and may approve or deny it based on whether the proposal has merit and the potential to fulfill all criteria for the Michelson Prize. If the Scientific Advisory Board approves the registration letter, the registrant signs the competition guidelines. This document is available for download on the prize application page of the website, and I recommend that you take a look through it at your convenience. The competition guidelines include information about the confidentiality of proposals, media rights, indemnification, and outline what is included in the master participation agreement that the registrar will sign in step 2. Additionally, at this stage, the registrant will speak to Found Animals General Counsel regarding the requirements of master participation agreement. The registrant now goes to work to develop the data needed for data package 1, and Dr. Johnson will discuss steps 2 and 3 and go into detail about the requirements for the 2 data packages.
Thank you, Katie. So step 2 will actually be creating the research information that will be given to our foundation to attempt to prove that you have the prize-winning process. And although our foundation has funded research proposals, we have not yet to date received any applications for the Michelson Prize in reproductive biology. So it's very important that any people viewing the webinar or accessing it on our website at a later date be sure to look at all of the criteria required in the prize application process. After the registrant decides to submit data for the prize, they first of all certify that they have all the data required for data package 1, and we'll go into this more in a bit. This will be the data that proves that their product works and that it's safe and that it's permanent. They will sign the master participation agreement, which Katie mentioned, and then they will submit the data to our foundation saying, this is what I have to show that I'm capable of winning the Michelson Prize in reproductive biology. They'll pay the application fee, and then their application will be date stamped. That will give us information on the earliest applicant that has the product that meets all of our requirements. The master participation agreement will include negotiation of intellectual property, and this may vary depending on whether the applicant is in, say, a private biotech firm or at a university. It may even vary by what product they propose, an injectable vaccine, for example, versus a cytotoxin, something like that. Our general counsel will negotiate with the registrant in the completion of this master participation agreement. And at that time, the investigator must certify that he or she has all of the data required for submission of this package prior to entering negotiations for MPA. This negotiation must not exceed 30 days. Now, for the next few slides, I want to talk about what the applicant must submit for data package 1 to give us some reasonable assurance that their product is in fact a permanent, safe, single-dose sterilant that can be given to both dogs and cats and last the life of the animal, as well as being of reasonable manufacturing price and of a product that could reach regulatory approval. So first of all, they must submit proof of concept data. That simply shows that it works. Katie mentioned that we require data from both rodent and target species. For us, the target species are dogs and cats. And the reason that we ask for rodent data is that mice and rats have a much shorter lifespan. So the applicant may be able to show lifetime suppression of reproduction in mice or rats, or at the very least show us some safety data over that period of their experiments. We also ask for preliminary safety. We will attempt to prove pivotal safety in later studies, but preliminarily we would like to see high dose effects in at least a 12-month rodent toxicology study where the treated rodents are monitored for body weight, food consumption, fecal elimination, behavior, growth, and histopathology. We also look for preliminary safety at the target dose, 1x dose being what we think we will give to animals if this product is approved, with enough numbers to demonstrate a statistical likelihood that the product will be safe in larger studies. We don't want to approve a product that some years later will cause adverse effects in our patients. We also require in Data Package 1 preliminary effectiveness. And what that will include is a blinded, meaning that whoever reads the data does not know if they're reading data from treated or control research animals. Blinded determination of a statistically significant effective dose in the target species for 1 to 3 years. Now, statistically significant is important because we want to use enough research animals that if there is a statistical difference, we determine it. But we also want to minimize the number of research animals that we use and not subject research animals unnecessarily to treatment or research protocols. The duration of following effectiveness will vary in time depending on mechanism of action. So, for example, a product that immediately causes total ablation of the testes and ovaries might be provable in 1 year, whereas something like a vaccine against a reproductive hormone may take several years to show that reproduction is not going to return. We recommend a minimum of 6 animals each per species and gender, treatment and control groups, again depending on the mechanism of action. Continuing in Data Package 1, more information that the applicant has to provide us as part of their application is a description of the pre-formulation composition. We want to know what this product is. Biochemically, we want to know information about our potential to manufacture this at a commercial scale and at a reasonable cost, how it will be packaged, how it will be administered, etc. We want a statistical justification of sample size in the data presented. We want quality assurance so that we know that the researcher has traceable and secure data records and that their equipment functions appropriately. And finally, we want the applicant to tell us what the likely mechanism of action is. It's not enough to say this product will make dogs and cats sterile. We want to know how that happens. What cells does it target? What mechanism does it use to induce the sterility? At the time we received Data Package 1, and the more detailed requirements for Data Package 1 are included on our website. At the time we received that information, Katie and I will do a preliminary evaluation of the data package for completeness, looking for those items you saw in the 2 previous slides. And if any of those elements are omitted, we will ask the investigator to provide whatever is needed to the foundation. The investigator will not get a date stamp, that is a stamp showing the date of their application, until all the sections are provided and the application fee is paid. If there are major omissions, the investigators must wait 2 months to reapply and must provide a new application fee, and again will not receive a date stamp until the resubmission is approved in terms of completeness and receipt of the application fee. After we determine that the data package is complete, the next step for the foundation is to review the data by a team of scientists competent in the area of the application. And again, this may vary by whether an application proposes a vaccine or a gene silencing tool or a cytotoxin. At that time, we will identify 5 scientists, members of our core scientific advisory board, or perhaps ad hoc members, to review the data in Data Package 1. And they will tell us whether, in their opinion, those data in fact meet the qualifying threshold for all criteria. Do the data statistically show that this is an effective and safe product? If it meets those qualifying thresholds, then we will send a site team to the laboratory of the applicant, including a person well-versed in quality assurance, to see that the applicant's laboratory has the capacity to have conducted the research reported in DP1. So, for example, you could not just make data up and send it in paper or electronic form and win the prize. You would have to show us where you did the research. We would track the equipment that you used, how you maintained your records, and assure our foundation of the quality of the laboratory before we proceed. As Katie mentioned earlier, it's very important to our foundation that the data must be acquired in research that is compliant with the foundation's animal welfare guidelines. And these, again, can be found on our website. They tend to be more stringent than many of the guidelines used, for example, at U.S. universities, where research is monitored through the IACUC process, through the Institutional Animal Care and Use Committee. We have some additional requirements, and it's important for people to look at those so that they can be compliant in the research they conduct. We will consider issuing exceptions on occasion if the animal research met other high standards but perhaps was not in compliance with a minor detail of ours. But we really would like to encourage applicants to be well versed in our animal welfare guidelines before they start generating data for data package 1. At that point, then, we will have received the application. We will have assured ourselves that it is complete. Our board will have reviewed the data to indicate that the applicant has met thresholds of permanent single-dose safety, applicable in a field setting, cost-effective, etc. And we will have assured ourselves through a site visit that the applicant comes from a legitimate laboratory where this research was performed. At that point, then, we will either officially approve or disapprove data package 1. If data package 1 is disapproved, down here at the bottom of this slide, the applicant will receive reviewer comments and may resubmit a new data package as soon as 60 days later with a new application fee. So, for example, if there were some statistical questions or perhaps not enough numbers for the safety data that we require, the applicant may want to continue on with this product but give us more information in an application. We hope that these DP1s will be approved because we're very excited about this research. We're excited about getting a product on the market, and we would really like to award the prize. So if DP1 is approved, then our next step is going to move into step 3. Remember, step 1 was the registration process. Step 2 was the application process, where the scientist or scientists generate data that they submit to us to try and prove to us that they have a product that will meet the prize. What step 3 is going to be is another research endeavor generating data for what we call data package 2, where we contract out to an external laboratory to confirm that this product really does meet the goals of the Michelson Prize in Reproductive Biology. And just as a quick aside, once we approve a data package 1, other potential applicants, and where would we find these? These would typically be people who have registered. They will be notified that a contender has been date stamped and is moving into step 3 of the process. And once we do our first date stamp, we are going to open a 1-year window trying to encourage other potential applicants to submit their data packages for consideration. The application window will allow us to consider proposals that we receive, say, over the course of a year, even after we've received the first one. And that is because we know that with the kind of research we're going to conduct to generate data package 2, that a lot of studies fail, at least studies by pharmaceutical companies who are trying to bring drugs to market. So we don't want to shut down interest in prize application after we receive the first application, or DP1. We'd like to encourage people to move into this step. And for the window, we will accept applications during that window and very likely proceed on to step 3 with subsequent applications. If the window closes and we've not received second or further proposals, we may even extend that window. So what is step 3? Step 3 involves a confirmation by our researchers that this product is in fact safe and effective and meets the goals of the Michelson Prize. To design the research that will be performed to generate data package 2, the applicant will submit a plan for the prize study design, and you're going to see in the next slides what we're going to ask in that design. We're going to consult in the research project based on what we want to know, and we are going to pay for the study to be performed in a contract research organization that performs its research using good laboratory practices to provide the final data to determine whether or not the product has won the Michelson Prize. There are 3 big parts to data package 2. One is development of the final formulation, because of course if you're going to test safety and efficacy in large numbers of dogs and cats, you really have to have the final product. You have to know the dose, you have to know how you will administer it, You have to know how you'll package it and store it. There will be a pivotal safety study, and this will position us to move forward with the Center for Veterinary Medicine at the Food and Drug Administration, because we believe this product will be a veterinary prescription item, toward regulatory approval. And there will also be what is called a non-pivotal effectiveness study in larger numbers of dogs and cats than we have required for data package 1 to show that this product does in fact work. Now, the first of these three pieces is the development of the final formulation, and this has to happen before we do any of the subsequent studies. So we have to look at scalability demonstrated by development of 1/10 of the size of the anticipated batch number that will be created when we commercially develop this product. So, and again, that will vary with what kind of product it is, a toxin, a vaccine, an IV gene silencing, viral vector, things of that nature. And for example, we will have to formulate and create 1,000 doses if we think the final batch size will be 10,000 doses. We will demonstrate at least 6 months stability of the formulated product. And as you remember from Katie's remarks, that would be either at room temperature or at refrigeration temperature. We will accept a product that requires what's called a cold chain or refrigeration during its course of storage. We prefer 2 years of stability, but at least 6 months of stability will be required. And finally, we will work with the applicant and our foundation people in developing preliminary packaging studies and cost estimate refinement. The second piece after formulation of the product is doing what's called the pivotal safety study. And this must include information on dose justification, which may include pharmacokinetic data. And these are things like, how is this product absorbed? Where does it go in the body once you inject it? How is it excreted? And also toxicological characterization, again, based on the evidence provided by the investigator in DP1. Is it likely to cause some damage to the liver or the kidneys? Or do we know that they are totally unaffected even by a high dose of administration? In the pivotal safety study, we will require at least 16 animals per group. That would be 4 male dogs and cats and 4 female dogs and cats each. And we will require for safety, and this tends to be standard for the industry, a dosing at 0x, that would be control, so they wouldn't get the product, but they might get the carrier or the injectable. 1x dose, which is the dose we think will be administered to our shelter animals if this product is approved, and 2 higher doses. And sometimes this is 2x and 5x or 5x and 10x, and that will depend a little bit on the character of the product. These doses are given to test animals, the 16 dogs and cats per group, for 12 months. And again, some details of this research will be determined by the foundation in collaboration with the applicant depending on the product. The protocol will be pre-approved by the Food and Drug Administration Center for Veterinary Medicine. And in their studies, it is a requirement that animals treated will be humanely euthanized at the study conclusion. And the reason for that is that all tissues need to be examined so that, for example, very subtle changes in organs that may be damaging but not obvious from looking at the animal could be detected. Now, the third part of step 3 is conducting of the non-pivotal effectiveness study. And for this part of DP2, we require a minimum of 10 to 15 animals per group. And remember, for animals, we mean male dogs and male cats and female dogs and female cats. So that translates to 40 to 60 animals at the 1x dose, the dose that we think will be given once the product is commercialized. And in this part of the study, the effectiveness study, we generally will require that treated animals be followed for a minimum of 3 years. Again, that will depend a little bit on what the product is, on how it creates sterility, and on whether there could be rebound or reversibility. Remember, in data package 1, the applicant tried to prove to us that in rodents, this is a product that suppresses reproduction for a lifetime, and it works pretty well short-term in dogs and cats. For this part of our work, step 3, the DP2 package, we want to follow pets for at least 3 years because our requirement for our prize is that it be a 10-year or lifetime effective product. This ideally will occur sequentially after the pivotal safety study. So if it turns out that the product is not safe, for example, we would not proceed into the effectiveness study. And that's it for Data Package 2. With those 3 elements of Data Package 2, we submit information to our Scientific Advisory Board reviewers, and they will make a decision either that the prize criteria were not met, maybe the product was not safe or it was not effective, in which case the applicant must wait at least 60 days before reapplying with an improved or different product. Ideally, results of Data Package 2 will show us that the criteria for the Michelson prize have been met. And if that's true and they have the first date stamp, they will be the winners to which our $25 million prize will be awarded. Finally, there is a third category wherein Data Package 2 information may show that the prize criteria are met, but there are other investigators currently in the Step 3 process generating Data Package 2 information. And if that's the case, unless the date stamp is the earliest, we will wait until the projects in Data Package 2 generation status are finished to see which ones do in fact meet the criteria and which don't. At that time, we will award the prize to the product that has the earliest date stamp. And that's our story. That is the prize application process for the Michelson Prize in Reproductive Biology. This webinar will be on our website at www.foundanimals.org. And the information in greater detail, for example, the contents of Data Package 1, are also accessible on our site. Now I'm going to turn this back over to Katie because we still have a few minutes before the webinar concludes, and we would be glad to field questions for anyone attending.
Thank you. Does anyone have any questions for us?
Hi there. I have a question. What is the status of the research currently funded by the Michelson Prize and Grants?
Great. As Katie mentioned, our foundation has about $50 million available in competitive funding, and we currently have approved and started the funding and research process in about 15 proposals, 2 of these outside of the United States and the others in the U.S. These tend to range from 1 to 3 year projects, and I believe we've committed about $6 million of our $50 million if we were to commit, again, sequential years or subsequent years funding. We're very excited about these projects. Our Scientific Advisory Board is good at finding good science, and I would say that probably at least half of these proposals may have the capacity to move on to a prize application. However, having said that, none of them are yet at the point where they've generated enough data for Data Package 1. So we're doing good work, we're funding good work, we're excited about the prospect that the prize will be awarded, but my personal view is we're probably still at least 5 years away from being able to tell you that the prize is on the horizon.
Well, thank you.
Thank you for your question. Are there any additional questions?
Yeah, I've got a question. Could you tell me some of the general approaches that people have been taking to do the sterilization without surgery?
Yes, I'd be glad to field that one too. This is a very tough challenge. The reproductive systems in all animals, from humans on down, certainly the dogs and cats, other mammals, even other non-mammals, are systems that nature and biology tend to conserve. So it's really tough to safely knock out reproduction. There's just a lot of kind of ancillary ways that the body tries to keep reproduction working. We have received proposals in 3 general categories of research to date. And in case I forget at the end, I just want you to know that we are really looking for other approaches as well, other innovative ways to stop reproduction. To date, we've had interest in 3 areas. One of them is generally referred to as immunocontraception, trying to create a vaccine so that the animal would make antibodies. But instead of making antibodies to like a flu virus or something that you would do with disease, you would make antibodies to reproductive proteins and keep them from having an impact in the body. So essentially you'd knock out the reproductive function. And when these work, they tend to keep, for example, dogs and cats even from going into heat, and we would find that desirable. The second category, actually the one we've received the most proposals on, is in the area of trying to selectively deliver a toxin to specific cells in the body to knock them out. And as an example, if you're a patient, a human patient with cancer, and you take chemotherapy, which is designed to kill the rapidly dividing cells of your cancer, sometimes they also kill the rapidly dividing cells of your gonads, of your testes or ovaries. And so people have theorized, you know, if we could get these chemotherapeutic agents and deliver them only to the testes, only to the ovaries, and knock out those reproducing cells without hurting the rest of the animal, that might be an approach. So we have a lot of talent around the world trying to develop carriers that will take toxins either to specific areas of the brain or specific areas of the gonads to knock that out. Then after immunocontraception and cytotoxins, the third major approach we've had is very interesting to me because it's the newest, and that's in the field of gene silencing. Work is in progress all over the world to try and turn off genes because we know that both canine and feline and human genome, and if we could turn off the genes that turn on the reproductive system, that start up in the brain and secrete proteins in the brain that start the whole reproductive cascade. If we could just cause those genes to shut down, it would seem like that would be a very cool and safe way to stop reproduction. So we have some work going on in that area. It's not simple. It requires delivering very discrete, bioengineered, interfering RNA to specific places in the brain. So it's not easy, but it's something that has a good potential. So those would be the 3 general categories. We also look hard, you know, if we could put in a microchip like the pet identification microchip that we use to bring homeless pets back to their owners, if there's some way we could put that in and it could beam a beam into the gonads to shut them down, or some cool bioengineering tool, or some other nifty way that nobody's thought about before, we would love to entertain proposals in different areas than those 3.
Thank you, Dr. Johnston. Are there any additional questions?
Can I find information on the members of the Scientific Advisory Board, the individuals who will be reviewing the contents of the data package?
Absolutely. We're very proud of our Scientific Advisory Board, and information about all our members is available on our website. You can also see biographies and photos of all of the members of the core Scientific Advisory Board, as well as our grantees. So I do encourage you to go on our website and take a look at all the additional resources that we have on there, including our resources page, which has a great canine and feline reproduction resource that was created by Dr. Shirley Johnston. That's a very helpful tool, as well as our quality assurance toolkit and additional presentations from the ACC&D Symposium on Contraceptive Resources. Thank you so much for your time. We really appreciate you joining us today, and please feel free to contact us if you have any additional questions.
Thank you.